Okay, so I did my presentation. Team two is doing their presentation on the ethical considerations of CRISPR and its validity to treat genetic disorders. But first of all, we need to understand what exactly CRISPR is. CRISPR stands for clustered regularly interspaced short palindromic repeats. That means it's groundbreaking gene editing technology. As you can see in this diagram on the right, the CRISPR here, you can see how the genomic DNA is making its way through the Cas9, and you can see how there's a matching sequence making its way into the middle of that gene that's broken down the middle, and it's inserting that sequence into it. CRISPR guides RNA in Cas9, which is a CRISPR-associated protein, to break the DNA, and it actually allows for the changes in the genome. That's right, we can alter genes with CRISPR. So, for the functions of CRISPR, we can edit the genome to prevent or treat genetic diseases. However, this is where the main point of this project comes in because it needs more research and testing on this aspect would make it truly viable. And other smaller heritable qualities could also be edited. CRISPR would also allow scientists to create much more accurate models of animals, and as such, accelerating very important research as cancer studies or mental illness prevention. Because if we can let scientists create more accurate models of animals, then since let's say for a rat, for example, a more accurate model of a rat means more accurate results when the scientist does a test on that rat, which would lead to better results for the scientific community as a whole. Now we'll go into the current uses of CRISPR. Currently, it's being used to test and treat single gene disorders such as Tay-Sachs, sickle cell, and cystic fibrosis. Again, these are majorly work in progress, but we're already in the testing process for these, and the hope is that we can actually cure them soon. Moving into viral disorders, we have chickenpox, measles, and Hermes, and then it's actually being used to help some cancers like leukemia and lymphoma. Now on the right, this diagram, we see how a single gene disorder is made, where the chromosome condenses then uncondenses, but you see that there's a green line in the middle of the blue chromosome. And that's what we call, that's basically a mutation. And that mutation in the chromosome codes for a different protein than intended. And that can cause things like cystic fibrosis. But what CRISPR can do is it can edit the gene, take out the green, and put in a blue, and then we can code for the right protein and solve. So that brings us to our hypothesis, which is that CRISPR-Cas9 is ethical, in fact, if we keep it restricted fairly, and it can treat genetic disorders if we invest research into it. First, we'll go over opinions of people with influence and knowledge, which is the entrance to our data section. So according to the National Institutes of Health, which is a government organization, there are indeed some biomedical concerns surrounding CRISPR. However, despite the concerns, this government organization seems to be okay with modifications to cancer cells or cells of hemophilia, especially since they have already done tests on it and have put out data on it, which I will be presenting later. Where the concerns come up is the difference between treating cancer and making your child look different based on how you want. Those are two completely different things, yet they are both within the reach of CRISPR. Judging by these opinions, the best option would be to limit what CRISPR can actually be used for to only threatening things such as genetic diseases and not convenience. And this is proven by the NIH themselves. This is one of the solutions they provided. Another source of information, researchers at Harvard propose a different point of view than the NIH. They say that with our current research, we can't determine whether or not CRISPR is ethical. Instead, we have to choose carefully based on more research done whether or not we want it in our society. And I do agree with this. We need to do more research into CRISPR. And that is why this hypothesis comes into play. So now I'll go over two different genetic disorders that could be treated by CRISPR. First up is albinism, which is a disorder that affects melanin production and severely reduces it. And this affects a multitude of things. First of all, it'll make your hair and skin much, much lighter to the point where they can actually turn white. And your eyes will have such poor cognitive function that you might as well be blind. Administering CRISPR to the eyes or to the skin. While the tests haven't been done on fixing the eyesight itself, and I don't think that would be true, it could definitely help get your hair color and skin color back to what it's supposed to be. As you see in this diagram, in the optic chiasm, you see how more fibers are crossed in people with albinism. This is due to a genetic mutation that could be solved with CRISPR. Next is sickle cell, which in fact is a just blood disorder that affects hemoglobin. As you can see in the diagram on the right, it's causing the blood cells to become a sickle shape because it's more, you see like the hemoglobin in the blood cell, it's becoming stiff and forming into the sickle cell. But again, CRISPR could help by replacing enough cells in the body to carry the sickle cell trait determined by alleles. Then we could introduce enough regular red blood cells into the body through CRISPR to help get rid of sickle cells. Now here's something very important, which is gRNA. gRNA is vital to CRISPR research, and you see the tedious effort that goes into gRNA testing, where you have to take a set, you have to rank it, then you have to filter it, and then if all of the genes aren't covered, you have to go all the way back to the ranking process. And you have to get a set of gRNA designs with the best possible design and coverage for CRISPR to work, proving why we need more research into this topic. So these are methods that have already been done before, and I'm going to share data on it with you, but testing CRISPR on models of rats and rabbits as well as on actual animals. But first, we need to understand some vocabulary for the data set, or else you won't, some people might not understand what it's saying. So a nuclease domain is what's responsible for the breaking of DNA. An effector and type are pretty straightforward. What it affects, what type of CRISPR it is. Track RNA is a type of RNA that interacts specifically with CRISPR and helps its growth. And PAM/PFS is used to mark sites where CRISPR is used. So here's the data set that was published by the NIH. So you can see the class of CRISPR on the left side, the type and subtype, as well as the effector and the target, which there's a multitude of targets, dsDNA, some ssRNA. And then you can also see the nucleus domain, what was used to break the DNA open for the CRISPR to go into it. As well as you can see that tracker RNA isn't actually required for CRISPR to function. As you can see, some say tracker RNA requirement no, some say yes. And then here on the very right are the sites where the CRISPR is used. So the ISCB bioinformatics team posted this diagram, which basically shows on the top left corner 4 samples of CRISPR and what's the end result was for that. There's the allele frequencies on the bottom left. There's deletion distribution on the top right, and on the bottom right there's SNP distribution for sgRNA. So here we see both limitations and advantages. So first we see the strategy the PubMed Central used was to deliver it through LV, as well as their delivery approach was through CRISPR-Cas9. And their limitation was that the vectors for the delivery strategy they used, which was LV, were actually at risk of mutations, which mutations are what CRISPR actually tries to fix in the first place. So this is a huge limitation that needs to be fixed with further research. But the advantage is that it can actually deliver CRISPR to lambda cells in single transfections with high cloning capacity. Another study published by them where they used inorganic nanoparticles to administer the CRISPR instead. But the limitations for this was slow degradation in the vivo and specific toxicity as well. However, the advantages were that they were small size, high effectiveness, and delayed controlled release, as well as much more. So as you can see by the previous pictures, while CRISPR is certainly effective, it does have its limitations. And these limitations include potential toxicity if an incompatible type of CRISPR is used, and the vectors risk mutation and degradation. This is why we need to the government needs to invest more research into CRISPR, and we need to convince the public that CRISPR is in fact ethical. Because if we don't convince them that, we are losing all this potential to cure genetic disorders. Because CRISPR is still a major work in progress, a lot of the mentioned benefits related to curing genetic diseases are not fully done yet. The major limitation is that our world's research into CRISPR isn't fully developed, causing various ethical considerations. And it's currently limited to models and animals too, since, well, testing it on actual humans is unethical. Although outside of the US, there have been tests on humans in China. However, those were deemed unethical, so we need to do further testing before it's deemed ethical on humans. So for the results for ethical considerations, after looking at the opinions from multiple government websites as well as scholars, we agree that CRISPR is ethical to an extent. The NIH put out a survey where it said that the public, a lot of it agreed that CRISPR was ethical, but there was also a major portion that did not like it because you are editing a person. Treatment for diseases is okay, but we shouldn't be modeling an ideal baby before they're born. The NIH knows this, and CRISPR is dangerous, but that doesn't change the fact that it's an amazing tool to use to combat diseases that previously were thought to be impossible to treat. As proven by the various data experiments on mice, rabbit models, etc., CRISPR is clearly valid to treat genetic disorders. We just need more research done into it and more funding. But also, moderation of CRISPR and what we use it for is key to convincing people that it is an ethical product. So as a conclusion, we absolutely need the government to recognize CRISPR. This is our solution. It can treat genetic disorders which we thought were impossible to cure before. All we need to get this research going is funding from the government and acceptance from the public. A mix of both technical and social aspects is necessary to make CRISPR successful. Possible solutions for this issue would be to educate people about CRISPR widespread and let people realize the potential of it. This is our solution to the ethics of CRISPR. So theoretical results, if our solution proves true, would be genetic diseases and disorders being cured, making for a much healthier world and much higher quality of life. Not only genetic disorders would be affected, but things like cancer would also be eradicated, especially seeing as we now have solutions like killer T cells. So here are acknowledgements and references.
Thank you, Manu, for that presentation.